All right, we'll just wait a few more moments and see if anybody else is joining us. Afternoon session is about ready to start. We seem to like to start right on time. It is 1:10. So welcome officially to the afternoon session of our 9th annual conference. We are glad to see you back. If you're here this morning, thank you for making your way back to the session. And if you were not able to attend this morning, we are glad you're here. Just in case there's some new people, just going to go through this small housekeeping list once again. As we did this morning, we just invite you to stay on mute as our speakers present their research. After their presentation, we will allow for questions once again. For questions, you may use the chat box that you should see at the bottom of your screen to type in your question. Or as we did this morning, if you would like to speak up and ask your questions by coming on video or at least on audio, you can certainly do that as well. We have some interesting speakers this afternoon, and I will pass this on to Cynthia Brink to move on with the introduction.
Thank you very much, Emily. And welcome to the afternoon session. We have a great lineup of speakers for this afternoon, and we've been really good with staying on time, and hopefully everything goes as planned. I would like to go over our land acknowledgement for our area. The Longpoint Biosphere region acknowledges that we meet on the traditional territories of the Adirondack, the Haudenosaunee, and Anishinaabe peoples, and show respect today to the communities of the Mississaugas of the Credit and to the 6 nations of the Grand River Territory, whose treaty lands include the Longpoint Basin region. This afternoon's first speaker is Amanda Loder. She is a PhD candidate at the University of Toronto in physical geography and is a member of the Paleoecology Laboratory. Her research focuses on carbon stocks in freshwater marsh soils and their capacity to sequester carbon and provide a means for mitigating the climate crisis. She also studies how land use and climate change affect, and the implications for wetlands, restoration, and conservation. Amanda enjoys working and engaging with wetland conservationists and is currently conducting research in partnership with the Nature Conservancy of Canada in Norfolk County. Welcome, Amanda.
Thank you for having me. I will proceed to share my screen now. Can everyone see that?
We sure can.
Excellent. Okay. Yeah, thank you for having me. I've participated in this conference the last few years, and I really enjoy seeing all the great research going on in the Longpoint region. So thank you to the organizers, and thank you for the land acknowledgement. Today, I will be presenting my doctoral research on carbon burial in natural and restored marsh soils in the Longpoint region. I am working under the supervision of Prof. Sarah Finkelstein, and would like to acknowledge that this work has been done with collaborators, as mentioned on the bottom right-hand corner of the slide. Wetlands contain significant global carbon pools and are known to affect the climate over long periods of time. Because they provide a low oxygen in wet environments, they can store and bury partially decayed organic matter that comes from plants, meaning that they can take up carbon from the atmosphere and sequester it via photosynthetic processes. Simultaneously, wetlands release methane, which is a potent greenhouse gas. When looking over hundreds to thousands of years on long time scale, the amount of carbon they take up and store in their soils outweighs the amount of carbon they release to the atmosphere, and thus they have a net cooling effect. Despite the significant role of wetlands in the global carbon cycles, many of their soil stocks are poorly constrained, particularly in the temperate region where large populations of humans dwell. One of these types of wetlands is freshwater marshes. Despite freshwater marshes not being well calculated for their carbon soil stocks, current research does suggest that they can have high rates of carbon burial in their soils. So on the box plot on the left-hand side here, we have short-term rates, which is mainly makes up the rates of carbon accumulation that have been calculated for freshwater marshes. And these are over rates over a couple of decades. When calculating these, various dating methods are used, including radioactive isotopes, stratigraphic pollen markers indicative of various increases in vegetation, as well as trace metals indicative of the rise of the industrial period. However, we have fewer calculations of rates of carbon accumulation over the long term, so over hundreds to thousands of years. Nonetheless, we do see a significant difference between short-term and long-term rates that have already been calculated. This difference has been poorly characterized. Suspicions of why we see this difference or reasons for this difference may be due to long-term decay that is accounted for in the long-term rates, so certain carbon fractions that don't get preserved in deeper soils, or the shorter rates may be elevated due to human activities, which could include higher biomass production from invasive species or nutrient enrichment. In southern Ontario, pre-settlement marshes were extensive prior to major landscape alterations. Over the last 100 to 200 years, a large proportion of freshwater marshes have been altered for agriculture and urbanization. Estimates suggest that the pre-settlement carbon stock of freshwater marshes is the highest of all wetland types in southern Ontario. But this carbon stock is highly uncertain because there's a lack of field data on total carbon stocks in marshes, because we don't have much information on the soil depths and their carbon contents. Because of the alteration that has occurred across southern Ontario, and because of more recognition of the importance of wetland functions, there have been major restoration efforts occurring across southern Ontario and elsewhere. In the Longpoint region, stakeholders, including the Nature Conservancy of Canada, who I've been working with, have been restoring wetlands. So in this map here on the right, the colored blocks are lands that are owned by the Nature Conservancy of Canada. And the dots are sites that have been restored. So many of these sites are actively restored, meaning that plants have been sowed and soils have been moved. Sites could also be passively restored, as shown at the site with the pink dot. And this is a means by which land has been abandoned, but natural wetland functions have been restored on their own without intervention. Most restoration projects have focused on increasing wildlife habitat. But because of the recognitions that wetlands can sequester carbon, there's been increasing interest in restoring wetlands as a means for providing carbon burial and as a natural climate change solution. But to this day, we still don't fully understand how well restored wetlands bury carbon and how this compares to their natural counterparts. Furthermore, existing research suggests that restored wetlands can be carbon sources in their early years, as the amount of carbon they release outweighs what they take in. But the time at which the amount of carbon they may take up and preserve in their soils, the time at which this occurs has a net cooling effect and outweighs the amount of carbon that's released is not really well known. So this switchover point is of interest for better understanding, to understand the role of restoration in climate change mitigation. So for my presentation today, I will be addressing these three questions. The first is what are the average short and long-term rates of organic carbon accumulation at a reference site? So meaning a marsh that has not been directly altered. The second is, does passive restoration in the Longpoint region provide a means for carbon sequestration? So carbon burial in soils. And the third question is, do organic carbon densities increase with age and actively restored wetlands? So this is the mass of carbon per unit areal in the soil. And by age and active restored wetlands, I mean within a chronosequence of a series of wetlands, do the older ones tend to have more organic carbon than the younger ones? So in the Longpoint region, pre-settlement marshes were extensive. With my colleagues at the Nature Conservancy of Canada, we were able to identify a reference site that has not been directly altered. And it is located in the central Big Creek Marsh, as shown by the red star. At this site, we collected a long sediment core that was 4.3 meters in depth. So this represents the sediments that have been deposited over the lifespan of this reference site. With this paleo record, we calculated short and long-term rates of carbon accumulation. So short meaning over the last few decades and long meaning over hundreds to thousands of years. Furthermore, we characterized the organic matter composition to see whether internal factors such as degradation, long-term carbon decay, are contributing to higher short-term rates in comparison to the long-term rates that we saw, that we see in the literature and that I showed earlier on. For the second and third part, so addressing the second and third research questions, we identified a passively restored site, which is located in the upper Big Creek Marsh. We also chose 14 actively restored sites across the region to sample for organic carbon densities. On the left panel here for A, this is an aerial photo of the passively restored site. And you can see to the right here, this is the remnants of the Big Creek Marsh. Using historical imagery, we were able to see that in 1954, this site was farmland. But in the 1964 image, 1954 for panel B and 1964 for panel C. So in 1964, we can see that vegetation has started to reestablish itself and soils were re-wetted. And so today, this site supports marsh functions and marsh vegetation. So this means that passive restoration likely occurred sometime between 1954 and 1964. At this site, we collected a 36 centimeter core and used this record to calculate short-term rates of carbon accumulation. So we use short-term in this for this site as it's only been restored for the past 50 to 60 years. Finally, we collected soil samples across the 14 actively restored sites and collected them down to 30 centimeters in depth for measuring the organic carbon density of the soil. So again, the mass of carbon per unit volume of soil. And on the map here, we can see that the sites are located across the region and more specifically located in the lower Big Creek block, the upper Big Creek block of the Big Creek watershed, as well as in Bacchus Woods. Okay, so I'm going to show some results for each of these questions. The first is the average short and long-term rates of carbon accumulation in a reference site. So coming back to Central Big Creek Marsh, as shown in the photo here. So on the right-hand side, we have a graph showing the sediments down to 4.3 meters. And the x-axis is the rates of organic carbon accumulation that we calculated using lead-210 and radiocarbon dating methods. We found that this site formed about 5,700 years ago. So this demonstrates, and from these rates of the carbon accumulation, we can also see that this site has been accumulating materials and organic carbon over thousands of years. In our calculations, we found that the average long-term rate and short-term rate of organic carbon accumulation are high and demonstrate high rates of carbon burial in the soils of Central Big Creek Marsh. But similar to what we see in the literature, we do see that the shorter term rates at the top of core are markedly higher. This graph is some data from our geochemical analyses. I'm not going to go into it in detail today, but what I want to show is that the different colors of the dots are clustered together and represent different sections of the core that went down 4.3 meters in depth. And so what we see here is change in organic matter composition as we go deeper in the soils. Particularly, we see a difference in organic matter composition for these blue, as these blue dots are clumped and represent samples taken within the top 50 to 100 centimeters of the soil. From these geochemical analysis to date, we've been able to conclude that the composition in the top of sediments is contributing to higher rates of carbon accumulation in the short term, and that some of this material gets degraded before it's able to be preserved in deeper soils. In short, this work demonstrates that we cannot extrapolate short-term rates to the long term. So over decades, we see high rates in these sites, but over hundreds to thousands of years, these rates are lower. Nonetheless, though, we see that rates of carbon burial in this freshwater marsh are high. Okay, for my second question, I'm going to be talking about the results we collected from the passively restored site. On the right-hand side, this is a graph showing the rates of carbon accumulation down to 36 centimeters in depth, as this core was shorter than at the reference site. For this site, we've been using metal analysis to reconstruct metal peaks from fallout associated with the industrial period. I'm also not going to go into this in detail, but to date, we've been working on constraining the lead peak associated with the peak usage of leaded gasoline. In our analysis, we have been able to estimate that the 23rd centimeter interval was likely sediment that was deposited around 1980. With this inference, we can calculate that the average short-term rate of carbon accumulation within the last 50 to 60 years since restoration of this site is fairly high, although it is lower than the rate that we calculated for the reference site. When looking at the deeper sediments here, we see quite a bit of variability, which may be associated with influxes of material when the passively restored site was re-wetted, perhaps from flooding from Big Creek. And finally, I'm going to talk about our data that we have collected from the actively restored wetlands. Okay, so this box plot shows organic carbon densities that were collected by site. So the sites being located across the Longpoint region. And these sites on the bottom are grouped by location of the watershed. So we have Bacchus Woods, some sites in Lower Big Creek, and then the last two at the end are within the Upper Big Creek. So we see a large range of organic carbon densities, despite these sites being located close together and despite similar approaches being taken to the restoration of these lands. But to address the main question about year, when looking closely at the year of each of these sites, so in the right-hand side here, showing that the lighter blue colors are sites that are older in age, darker blue, younger in age. We see that organic carbon densities in their soils do not increase with age. And we don't see a trend that the newly restored sites consistently have lower organic carbon densities than the older sites. So we had expected that these densities would increase and that when a site becomes restored and becomes wet again, vegetation reestablishes and would result in biomass production and burial of organic matter and carbon stores in their soils. As a result, we are now looking at other factors to determine what are better predictors for organic carbon in the actively restored sites. So some of these factors include the slope of a given site, its position within the watershed, hydrology, so whether it is influenced by groundwater, and surface soil. So in this image here, this is a soil layer that has been produced by Agriculture and Agri-Food Canada, and and it demonstrates soil characteristics for the top few hundred centimeters. And we've been able to collect some data we've been able to extract data, I should say, to determine what are some of the underlying soils at the actively restored sites. And looking at the position of the sites, we see that some of the sites are located on soils that are sandier and better drained, whereas other sites are located on soils that aren't as well drained and contain more silt and clay. So next steps will involve looking more closely at these substrates and seeing whether this has an impact on whether an actively restored site has higher organic carbon densities within the first 10 years of restoration. In conclusion, we found that Central Big Creek Marsh has buried carbon at high rates in soils and has preserved carbon over hundreds to thousands of years. And so conservation of these sites is important as we're protecting ecosystems that have existed and established for long periods of time. In the passively restored site, we did see high rates of carbon burial, particularly when the site was first restored. In the actively restored sites, organic carbon densities did not increase with age, meaning that age is not a significant predictor of organic carbon stocks in actively restored sites within 5 years since restoration. But we did see high variability across the Longpoint region. So the implications for conservation include a need to consider deep carbon stocks in natural marshes. So when doing carbon inventories and trying to calculate the amount of carbon in wetland soils, it's important that we don't just look at the top 50 to 100 centimeters, which has often been done for freshwater marshes. It's important to go to the deeper stocks as we could be missing important carbon contents. The second point is that it's important to distinguish timescales over which carbon is buried for carbon accounting purposes. So we find that rates of carbon accumulation are quite different over decades in comparison to what they are over longer periods of time. And this is important when understanding their potential to play a role in climate change mitigation. And finally, other factors are more important than age and actively restored site, and it may be important to take a landscape approach when assessing how well actively restored wetlands can regain their soil carbon stocks. Thank you for listening to my presentation, and I would like to acknowledge those that have helped me tremendously with this work. I've been working with the Nature Conservancy of Canada, and Liv Monkwip and Brett Norman have been a huge help with this research. So thank you to both of them. And I also would like to specifically mention all of my lab mates who have helped me with the field work and other aspects of the research. And with that, I would be happy to take any questions.
Thank you so much, Amanda. Love to hear about your research, and the floor is open for questions. So feel free to use the chat box or lift your hand or unmute yourself if you have a question. Great presentation. You put a lot of details on there. I guess it's.
Oh, thank you.
Cindy says, great, timely subject.
Thank you. Realize everyone's just had lunch, so maybe we're still digesting and getting kind of into the science and all the great research again.
Yeah, and you see Amanda's email on that last screen there. If you have some question, I'm sure she will welcome them as well.
I have a question.
Jana, go ahead.
All right. Yeah, I was just curious how sort of going forward, if you plan on expanding on this, or what you think you or someone else could do to sort of increase the scope of this research, because it's, like someone else said, it's timely and important.
Yeah, for sure. So I'd say one of the efforts and initiatives that have been going on more recently is just quantifying the carbon soil stock in wetlands across Canada and better mapping out the wetlands, not only in the temperate region where we are, but also at more northern latitudes. So because we don't fully know what carbon stock exists in our soils, and understanding, if we go and alter these wetlands, what are we releasing to the atmosphere, and how does that contribute to our national greenhouse gas emissions? So I'd say that's a broader picture when thinking of wetlands that currently exist. In terms of the restoration work, I do see next steps would be looking at directly the greenhouse gas fluxes that come from the sites. So looking at how much carbon they may be emitting and trying to better understand how that compares to what we see currently in the soils. It does appear, though, that within 10 years of active restoration, that the soil carbon may not accumulate as fast as we think, and of course 10 years for us seems like a long period of time, but for an ecosystem, that's not necessary. That's actually a very short period of time. These are very young systems. So I would say the greenhouse gas component is quite important, and also important to match up with what we see in what is preserved in the soils.
So you need to take one of those McMaster flux towers and drag it over to wetland from the forest.
Yeah. Oh, pardon.
No, I don't know if you were there's the flux towers that are nearby, but in forests.
Yes.
If you could drag one over to a wetland, then.
Exactly. Actually, I do have a colleague at the Ontario Ministry of Natural Resources, and that group is currently working with the Nature Conservancy of Canada to set up a flux tower at a marsh.
That's great.
I do see two questions that have come in the chat. I'm mindful of time, but can quickly answer at least the first one. Did you find that soil type affected the amount of carbon you found? That is something I'm currently working on with the actively restored sites to see if the substrate affects it. And I noticed that someone mentioned the newly restored good as the older sites. And yes, of course, wetlands provide multiple benefits, wildlife habitat, other processes. So really this research, we're trying to, understand the extent to which these sites sequester carbon and what that means for regional and global carbon budgets. But yeah, the conservation and, trying to restore sites so that we can protect what's already stored in the ground is also important.
Thank you so much, Amanda. Wonderful presentation. A lot of great questions. One last comment, I'm sure you're seeing that as well from Betty Cheney. Terrific presentation, really enjoyed, important information to me. Thanks, Betty. I'd like to introduce to our next presenter, Victoria Tawa is a master's student at McGill University and the Red Path Museum in Montreal, Quebec. Having completed a bachelor's in biology with a specialization in ecology, Victoria now studies under the supervision of herpetologist Dr. David Green and inspires to study the ecology and conservation of amphibians and reptiles for as long as Dr. Green has. That's a large compliment. Victoria and her lab mates spent the past summer in Longpoint conducting field work for various projects, all in the hopes to monitor and to better understand and to inform future endeavors to conserve the unique fauna, especially the amphibians that call Longpoint region their home. Welcome, Victoria.
Hi, do you guys hear and see the presentation?
Yes, we do.
All right, great. Thank you, Cynthia, for the introduction. I'm really happy to be here and to be showing you guys the start of my master's research, what's been going on so far with that. So Victoria, and I'm going to talk to you guys today about the responses of a lakeside anurine community, so that's frogs and toads, to natural and anthropogenic disturbance. All right. So when we typically hear that term disturbance, we automatically tend to think of that negative connotation, right? And those negative effects of disturbance. But in reality, disturbance can actually be positive for some species. And so it changes the environmental conditions by altering resource availability. So it changes what species of resources are there and all that sort of thing, and can return ecosystems to earlier successional stages. And so that is actually a positive for species that are adapted to those types of ecosystems. Typically, a disturbance has been studied on a relatively small scale because it's pretty complex to look at all the responses of biota and all that. So we're really lacking a lot of ecologically relevant data on disturbance in general and animals' responses to that. And so that's kind of where my research comes in. I wanted to study the responses of amphibians, anurines specifically, to a bunch of disturbances. And so Longpoint was actually the perfect place to do that because, well, here's a picture of Longpoint. As you guys know, it's beautiful. It's a sand spit that juts into Lake Erie. And although idyllic and beautiful from this drone shot, we can see even here that the landscape is plagued by this invasive reed, the famous frag. And so there's been a lot of effort to get rid of the frag. And so that in itself is a disturbance where we applied a herbicide, we meaning a lot of organizations have done so. And specifically the Longpoint Provincial Park and the NWA, the Thoroughfare Unit of Longpoint, was sprayed in 2020 with a Roundup and a spreader that was aquatic safe. And so that in itself is a disturbance that is potentially affecting the amphibians in the area. And another disturbance actually comes forth in the form of a dune washout. And in this case, we can see in this picture A right here, this is the dune that was previously there in 2019. And here you can see the Lake Erie is right there. And the dune is preventing the lake water from breaching into the wetland. But by 2020 in February, the lake water had risen so high that it had completely eroded the dunes and water had gone into the marshland. And here in this last panel in C, you can see the dune is completely gone. And so by June 2021, when I was there, you can see this complete landscape change with the sand completely all the way into the marshland now. And so really the landscape and the spraying of the herbicide kind of made a study design for me. There were areas that were affected by that dune washout that I just showed you. Sprayed by the herbicide, spots only sprayed by the herbicide, and then spots affected by both of those disturbances. And then of course, there were also places that were not affected by either. And so that really made a good control. In addition, I also had access to a before type of temporal design. So I can use data from August 2020. We did a survey back then and can compare it to data that was collected in 2021. And so this is just the study site kind of mapped out in a picture. Here you can see the green is where the frag was treated, where it was sprayed either aerially or on the ground. And then the dotted area, it's a little less clear in this small photo, but the dotted area is where the frag still was, and at least as of 2018. And then you can see here this brown layer that I added is where I mapped out the dune washout, so the extent of the sand that has gotten into the marsh. And then all the dots are my sites. So just for visualization, so we're all on the same page, this is an example of one of my control sites. This is just two pictures, just so temporal difference between the seasons. So this is at the beginning of the season, and this is more at the end of the season. And here, neither disturbance was affecting this site. Here is an example of a dune washout site. And yeah, you can see the sand all fully into the marshland. Here is an example of a site that was sprayed with the herbicide. Here you can even see the tracks of the marsh master that came in after it was sprayed to squish down the frag to help it to decompose. And lastly, here is an example of a herbicide and dune washout site. And you can't really see any evidence of the spray because that was probably a ground spraying there. But I assure you, both disturbances were affecting here. So that brings us to the methods that I used. And in order to catch adult frogs, tadpoles, and fish, I used minnow traps. So I placed 5 minnow traps per site. These are minnow traps here. I placed 5 per site, and they're all placed among the emergent vegetation and checked approximately every 12 hours or so. And so that led to about 10 trap days per week. And I added these pool noodle floats, as you can see here, inventive little placement, but they were meant to add an air pocket so that if I caught any air breathers like adult frogs, and even we caught a few snakes, so that gave those air breathers a chance before we emptied the traps to get air so they didn't drown in the traps. I also had some visual surveys where I looked around for amphibians, and acoustic surveys where I listened for the males calling, just to increase the probability of detection, because amphibians are famously and notoriously bad for detecting. I also have some environmental data, and that's kind of the preliminary results that I'll be presenting today. So one of the hypotheses that I had for what might happen when the herbicide was sprayed was that because the glyphosate, which is part of the herbicide, kills the frag, but also algae, and then the frag is knocked over and left to decompose, there would be a bit more plant matter to be decomposed in those sites where the herbicide was sprayed. And plant matter decomposition needs to be done by aerobic bacteria. And so aerobic bacteria need to use oxygen to do their job. And so as they use more and more oxygen, there's a potential that they will decrease the oxygen to levels that are less favorable for the existence of other species in the water. And so I predicted that there's a possibility that there might be a decrease in frog and toad occupancy and maybe abundance in those sites that were affected by the herbicide. So I wanted to know essentially did the increase in plant matter decomposition actually decrease the amount of oxygen And did that translate to a decrease in anurine occupancy in those sites? So is that what's actually happening, or are these ponds buffered by some other factor? And so these are just some preliminary results. They're not beautiful yet, but they will get there. Here I have the dune washout sites versus non-washed out sites, and the yellow is washout site. And you can see there's a higher percent oxygen in the water in a site that was in sites that were affected by the dune washout. And this is where the hypothesis comes in, the herbicide sprayed versus not sprayed. It was actually opposite to what I expected. I expected that the herbicide sprayed ponds would have lower oxygen because of the increased plant matter decomposition, but it actually had higher oxygen. And that kind of puzzled me for a bit. I wasn't really sure what happened there. So I looked a little deeper and separated each site on its own. And here you can see it's this site type right here, the one that's affected by both disturbances, that's actually driving the results of the other two graphs. And so in here, the control is not different from the herbicide effect, and the control is not different from the dune washout. And it's really just this two disturbance site that's different from the others. And so that's still a little confusing to me. So I decided to look at something that was a little more general, and that's the oxidation reduction potential. And so ORP, oxidation reduction potential, is essentially the ability of a water body, for a water body to cleanse itself and essentially break down plant matter, waste, contaminants, and that sort of stuff. So this measurement looks at oxygen, but it also looks at chemicals that are functionally similar in redox reactions. So in here, we can see there's a higher redox potential in ponds that had a dune washout than ones that were not, versus here in the herbicide sprayed versus not sprayed, there was no difference. And so looking down at that further, again, it's even more clear. Any site that was affected by the dune washout had a higher mVORP. And a higher value means typically a healthier pond. It's able to decompose dead tissue and get rid of any contaminants that could destabilize it. So we're aiming for better, healthier ponds. And so any site that was affected by that dune washout is what exhibited the higher ORP values, irrelevant of whether it was sprayed or not. And so that really made me think that, maybe this dune washout is buffering the effects of removing the Phragmites. And so that's an important potential result, because I'm sure many of us have heard of these cuties, these little grumpy men. This is the endangered Fowlers Toads. Their northern populations are located only in Ontario, and it's a habitat specialist. So they're very specific in what they need. They need early successional habitats, well-drained soils, and they need to breed in shallow pools that are clear and relatively low nutrients. And previously, it's been shown that an important predictor of their decline was actually Phragmites. So I wanted to see, given these disturbances, where was it that the toads actually chose to breed? And these guys, as you can see here, I've mapped out where all the calling males were. So we did our acoustic surveys and listened for all the males calling. And here it's just, I'm going to zoom in here. There we go. This is the area where the dune washout was occurring. And you can see that these toads mostly called at the edges of where that dune washout was happening. And here I've circled just where we actually saw successful breeding. And that was in the form of either eggs, tadpoles, or a mating pair. And if we look even closer, we can see that these sites are the dune washout sites and the dune washout and herbicide sites. Those are also the areas that we considered to be healthier ponds, at least in regards to oxidation reduction potential. And so it seems like, which was expected, that they preferentially chose sites that were affected by the dune washout, because that's the kind of habitat that they like, the sandy bottom pools, shallow water, etc. But they didn't seem to avoid sites that were treated by the herbicide. They called and bred in those sites like normal, just like they would in the regular dune washout sites. And so I can't say this statistically yet. I still have some data to analyze, but observationally, at least we can look at the map and say it's very likely that the herbicide didn't actually negatively affect the Fowlers toads at all. So in regards to this project, the next steps are to really look at that, to be able to say that concretely, I guess. And I also want to look at whether, because they had such a great year this year, the Fowlers toads, I wanted to see their population growth, but also their individual growth compared to previous years. Since we have such an extensive data set, since David has such an extensive data set from previous years, I'd love to compare that to last year and see if there's any difference between their individual growth rates as well. And then of course, it's to finish IDing the individuals that I actually caught in the minnow traps, including the fish and other species of anurans, because there was over 300 photos each time we went out. So it's been taking some time. This way I can apply some before after control impact stats, and I can actually concretely say if there were any occupancy differences between these site types for not just Fowlers, but for other species as well. So my hypotheses were kind of in regards to habitat preference for this. So that was more sandy bottom pools that were produced by the washout are ideal for Fowlers, but species like American bullfrogs need those deeper ponds to breed in. So I would say that they would not be benefited by the washout. So just quickly to summarize, we have our two types of disturbance, and in both cases, the environment was altered. And so I'm looking at what type of site species occupancy changes will have occurred, and then how that relates to the species' habitat suitability and their preference. And so the implications of this research then is to potentially reveal some indirect impacts of the environmental management methods that we've used. But it's pretty interesting to see that these disturbances might actually have benefited the Fowlers toads. So overall, I hope that this research will contribute to a better understanding of the potential interactive or potentially additive effects of disturbance in a natural field setting. So thank you all for listening. I appreciate it. And I'd just like to thank the people who have helped to facilitate this research. Dr. David Green, of course, a bunch of others, and my lab mates and any volunteers that I've had so far. So thank you.
Thank you so much, Victoria. It was an awesome presentation. We've got 5 minutes for questions for Victoria. So you can either put your mics on and ask your questions directly or put them in the chat box for her. Michael McTavish.
Hi again. Thanks, Victoria, for the presentation. Really, really interesting. Great to hear about that. And also having worked in the area, it's just really fills in the bigger picture, which is great. You focused a lot on sort of physical and chemical aspects of habitat suitability, I would say, and I'm curious if you have any thoughts about other things in particular. I think sort of food availability. I mean, we work on insect communities in Phragmites, for example. We sort of think of it as this depauperate area, but there are a lot of insects there, sort of a different kind of community that could be potential food resources, but then also changes in vegetation. I mean, in Longpoint, I think after all this great success with the herbicide application, one of the big questions going forward is kind of what comes back from a vegetation standpoint after that, and it can sort of fall into lots of different vegetation communities. So I'm wondering, maybe it's beyond the time scope in particular of the masters, but I'm curious if you have some thoughts about those aspects of habitat.
So I did take some photos so that I'm able to ID. Are you able to hear me? Yeah, okay. Sorry, I was able to take some photos of plant species that did come back in those herbicide sprayed sites. And that's part of the IDing I still need to do. But yeah, I'm looking at how that will, I'm just going to include it as a factor in my analyses to just control for it, just differences in plants. And when it comes to insects, these, for example, the fowlers, they breed outside of these ponds, not breed, sorry, they feed outside of these ponds. They feed on the beach on the outside. So that was less important, but we do end up having a lot of dragonfly larvae in our minnow traps, and they tend to predate on tadpoles, but as adults, they are instead prey. So that's definitely a factor in there as well that I'm going to include as much as I can.
Great, thanks very much.
Great question. Kathy's got a question for you. Well, she's got a comment actually. So great to hear some good news about Mother Nature healing. So true. It's good to hear the good news stories. And I did see Jonah's hand go up. Jonah?
Yeah, I'm just gonna go along with Michael's thing and ask. And again, like we all know, there's only so much you have time to do, and more looking to the future. Light and sound issues, is that something you've considered or would consider for affecting these species, like noise pollution and light pollution?
I would say I probably could include that as a year thing. So in 2020, I'm really comparing 2020 to 2021 for the most of this. And in 2020, the park, Longpoint Provincial Park, was actually closed because of COVID. And in 2021, it reopened, and there's a lot of campers. There's a lot of people walking around. So I could probably consider that as a year difference, something I could consider statistically rather. But in regards to the NWA, a lot of people don't walk there because you're not supposed to stop and sit anywhere on that beach. So there's very few people actually on the National Wildlife Area. And that's where the majority of my sites were. So in regards to light pollution, there's not too much there.
Thank you.
No problem.
Wonderful. Thank you very much, Victoria. That was an awesome presentation.
Thank you.
Now we'll move on to our next research by Ian Fife. Ian Fife is the Forest Birds Program Coordinator at Birds Canada in Port Rowan. He is chair of the Forest and Treed Swamps Working Group, which he will be speaking on behalf of today. Ian graduated from Trent University with a master's in avian ecology, researching forestry-related effects on breeding birds. Ian has worked with birds across North America in various ecosystems, from Carolinian forest to the Arctic. Today, he will be talking about some of the conservation actions the Forest and Treed Swamps Working Group are applying to protect, grow the forest areas in and among the Longpoint Biosphere region. Now, I know that you wanted to do a sound check, Ian.
Hi, can you hear me?
Yep, we sure can.
Awesome, great.
Go ahead, you got the.
Actually, I don't have my camera on, sorry.
That's okay.
I'll just go ahead. I'm not going to fool around technology, a time crunch here. So good day, everyone. As Cynthia mentioned, my name is Ian Fife. I'm the chair of the Forest and Treed Swamp Working Group, but my primary position is with Birds Canada as the Forest Birds Program Coordinator. So this afternoon, I'm going to discuss a bit about the Forest and Treed Swamp Working Group, how it came about, what conservation priorities we felt were important for the Longpoint Walsingham Forest Priority Place. I'll end with a brief summary of the current on-the-ground actions that the working group is currently undertaking. So Environment and Climate Change Canada identified various areas across Canada that have important significance to species at risk and species diversity, or they just class areas that required significant environmental action. So in Ontario, ECCC selected an 86,000 hectare area of Longpoint and inland Norfolk County that best represented these important conservation initiatives and identified the area as the Longpoint Walsingham Forest Priority Place. I'll be using the acronym LPWF throughout the presentation. So ECCC approached environmental organizations, provincial and municipal governments, private and public landowners, as well as industry within the LPWF in 2018. And that was to identify the most pressing threats that require the most attention. They invited 23 local organizations to develop an integrated conservation action team. And they all employed science-based adaptive management framework of the open standards for the practice of conservation. And this led to the Integrated Conservation Action Plan, or ICAP, which is a place-based approach to conservation. Among the open standards framework, and based on the expertise and influence of the local organizations, many of the threats were identified through a situation analysis. So the situation analysis just represents the current conservation targets and threats, but it also takes into account the social, economic, biological, as well as the political systems that may affect local biodiversity. Well, the number of threats were narrowed down to 5 as the most important threats that needed to be addressed. And the Forest and Treat Swamp Working Group are focused on number 5 here of logging and wood harvesting. Within the first iteration of the situation analysis, the forest and treat swamps in the LPWF rated in good condition. However, a large portion of the priority places species at risk and biodiversity reside in our forested ecosystems. Also, climate change appears to be having a greater impact on ecological features such as vernal pools and ephemeral wetlands, in which many of our at-risk amphibians inhabit. So a reassessment of the forest and treat swamps had updated the condition from good to fair, and then a working group was formed based on that reassessment. There were 2 key ecological factors that were identified that we felt would benefit the LPWF forests. And so those objectives are to increase connectivity between our forest patches, as well as increase the amount of forest interior, which is defined as the amount of forested area that is 100 meters from a forest edge. So both of these objectives create habitat that allows flora and fauna to disperse freely. It increases biodiversity throughout the LPWF, as well as increases habitat for our forest interior species. So the 2 direct threats the Forest and Treat Swamp Working Group identified that we felt impeded these ecological factors were unsustainable forest management practices and invasive species. So I'll say invasive species a lot in this presentation, so please be ready for that, especially if you have an aversion to the word, maybe just the plants. But anyway, so the 2 direct threats act as kind of an umbrella for contributing factors. So the first main threat of unsustainable forest management is reflected in silvicultural practices such as high grading lumber. So what this means is that they are taking the best quality trees. So this reduces good genetic stock of trees, it reduces tree diversity, and it also prolongs tree diseases. The other unsustainable harvesting practice that takes place in our area is diameter limit harvesting. And this is the practice of taking the biggest and largest trees only. Again, this reduces genetic stock of our trees. It reduces seed production, so regeneration is slower. And over time, this creates a forest with reduced quality and smaller trees. So the following I'll address here are contributing factors that lead to the threat of unsustainable forest management, and followed by why these factors are important to conserving and increasing forest habitat. So there is a lack of knowledge of interior forest cover and especially connectivity within the priority place. So the working group is developing a model to identify areas that create the greatest barriers to wildlife, and this factor will be treated through tree planting initiatives. There's also a lack of best management practice implementation for species at risk. This factor has been a continuing roadblock no matter where you go and where wood harvesting is concerned. So Birds Canada is taking a step forward to identify ways that both habitat protection and wood harvesting can find a balance. And the other factor among this is retaining some of those trees that have died from invasive species such as emerald ash borer or beech bark disease. While tree salvaging is a good way not to kill live trees, there are also many advantages to allow these dead trees to remain on the landscape, not just as cavity trees, but the retention supports overall forest health from providing soil nutrients to wildlife shelter. And finally are some of the ecological factors surrounding poor harvesting practices. And these can result in younger and less diverse forests. This brings us back to the high grading trees for lumber and diameter limit harvesting I mentioned a moment ago. Additionally, our winters are warming. And so that means there's an increase of soil compaction, soil disturbance, and soil erosion that just provides an environment for invasive species to take hold. So the second threat of invasive species is founded in a lack of education and awareness as a contributing factor. Is a contributing factor of a threat to our forests and treat swamps. So sticking with the poor harvesting practice theme for a moment, recreational and harvesting vehicles can carry seeds from one area to another. This increases the spread of invasive species. So bringing awareness to washing vehicles before they enter an area would help alleviate this issue. Also, yard and garbage dumping not only makes our area look unpleasant, but it also increases the spread of invasives. So again, education and awareness and encouragement for planting native plants can also reduce the spread of invasive species. There are also some barriers to reducing invasive species that primarily has to do with the costs involved with having them removed. Additionally, invasive species are perpetuated on private land because mitigation efforts are not as accessible for private landowners. So who are we and what do we do? So there are 8 partners among the Forest and Tree Swamp Working Group. We're still in our first year of our action plan, but each organization are making impressive advances to protect and increase forest cover and connectivity, as well as reduce the spread of invasive species within the LPWF priority place. The first organization is ALICE. So in collaboration with Longpoint Region Conservation Authority, they have planted more than 18,000 trees, which amounts to 30 acres of reforestation efforts on farms across the LPWF. These projects include windbreaks and wetland buffers, and that aids in our connectivity between those forest patches, as well as upland block planting, which will increase our interior forest habitat. Alice is also engaged with the Norfolk Environmental Stewardship Team, which is a summer program of post-secondary students seeking careers in the environmental field, and they aided Alice in tree planting efforts. So the upcoming goals in the spring of 2022 for Alice are to plant 40 acres, of which 20 acres are already queued to be planted. So within the Forest and Tree Swamp Working Group, Birds Canada role is slightly different because we aren't land managers like the other organizations I'll be talking about. Excuse me. Instead, our role among the group is for species at risk detection and habitat protection. So for the past decade, we had monitored 4 species at risk, with the red-headed woodpecker added just last year. The numbers of our target species from 2021 are consistent compared to the last few years, the exception being for prothonotary warbler. This is not because their population has declined. We rely on volunteer activity, and with COVID, that activity slowed down. I'm still confident that the numbers remain pretty consistent in Bacchus Woods, where the majority of their population is in the LPWF priority place. In fact, the total number of prothonotary warblers detected across the Ontario range has increased substantially over the past few years. So this is some very good news for probably one of Canada's most endangered birds. So our detection surveys are then used to engage private and public landowners, as well as forestry industry, to encourage species at risk habitat protection. The other project Birds Canada is leading is monitoring the water levels of treed wetlands. There has been some anecdotal evidence from naturalists, experts, and professionals from the area that have noticed our treed wetlands have been drying out earlier in the spring at critical periods for amphibian and bird life cycles. So the objective of this project is an attempt to determine if climate change, drainage systems, or a combination of both are contributing to the increased speed at which wetlands may be drying. Obviously, we won't be able to measure when the wetlands were drying out in the past, but hopefully this will be the beginning of a long-term project. And for the purpose of this group over the next few years, we are hoping to be able to determine if drainage systems are playing a part in treat swamp water levels. So we're very excited to see some of the results from the first year of these data loggers. We just removed them over the last couple weeks, so hopefully we'll have some data to present soon. The next organization is the Longpoint Basin Land Trust. They're developing site-specific habitat management and restoration plans, and with the intention of taking pine plantations and returning them to their more natural, diverse conditions, as well as controlling invasive species on land trust properties. So they are improving forest habitat. They're also improving forest habitat by closing and preventing unauthorized trail access to species at risk and sensitive habitat, as well as posting and erecting signs to promote passive recreational use only. Unfortunately, unauthorized vehicle or recreational use is too common an activity in our forested areas, and our treed wetlands really take the brunt of that, especially in the spring when wetlands are the wettest. There are major impediments to these. These are major impediments to our ecosystems. So, and as well as the diverse species that rely on this habitat for breeding and caring for their young. Another major focus for the Longpoint Basin Land Trust is improving their sites through mechanical and chemical control of invasive species. One particular way the land trusts are reducing the spread from passive recreational users is installing boot brushes stations at trailheads on their properties. And here we see the land trust program manager, Kristen Richardson, demonstrating the boot brush at the Arthur Langford Nature Reserve. Our next organization is Longpoint Region Conservation Authority. Their focus is also on invasive species control. Apart from their collaboration with Alice for tree planting, LPRCA have mapped 20 properties for a total of 157 acres that are ready to be treated for invasive species. They also controlled LDD moth, formerly known as gypsy moth, on 13 properties this past spring. LPRCA also partner with private contractors and have species at risk surveys completed on 88 acres of land. I'd also like to add that Birds Canada is a partner with LPRCA as well. This past year, we surveyed approximately 4,200 acres of LPRCA property, targeting the species at risk I mentioned previously. So the Longpoint World Biosphere Reserve is another partner. It's been pivotal for just all 5 working groups, not just the Forest to Treat Swamp Working Group, in outreach engagement with the general public about the priority place. So the Biosphere Reserve printed just over 70,000 newspaper article inserts. Just over 45,000 inserts were delivered to the people of Norfolk County. The remaining inserts were distributed to various tourist provincial park locations this past summer, and any remaining inserts will be delivered next spring. They're also the Biosphere Reserve is also very active on social media, as well as producing blog posts. And this has included interviews with local scientists on addressing climate change issues. And also, we can't forget the outreach and education involved with a conference like the one we're all here today attending. So the next group or partner is the Nature Conservancy of Canada, and their focus is on invasive species management and control as well. This past year was their 8th year of landscape scale herbaceous invasive species control. Yeah, so in forested systems, NCC have a particular focus on garlic mustard. For those who aren't aware, garlic mustard displaces native vegetation. It reduces plant diversity. It creates a monoculture forest understory, which you can see in the top photo here. They also take a strategic approach to controlling woodland invasive species. So they target along forest edge habitat, which is often where the large host populations of these plants are creating a large seed source that contributes to this further spread into our forested habitats. Bacchus Woods is one of the best remaining examples of Carolinian forests and home to many species at risk. So it is a priority focused property for this control work. This past year, NCC treated 98 hectares of garlic mustard across 54 properties. The staff, NCC staff, will analyze data from this year's control efforts to determine the effectiveness of the treatment, as well as assess impacts of previous treatments on the population sizes and densities. and this will inform next year's control work. It's also really exciting to hear that several previously undocumented populations of spotted wintergreen, which is a threatened plant in Ontario and seen in the bottom photo here, were observed during the control work. So removing these invasive species allowed our native plants to come in, species at risk to come in. So that's fantastic, great news. So that our next organization is the St. Williams Conservation Reserve. They're focused on two contributing factors to increase forest health. That's recreational management and the protection and enhancement of species at risk and their habitat. So under recreational management, the Conservation Reserve have been collecting data on the type and frequency of recreational use on their lands. Similar to the Land Trust, they are monitoring compliance with recreational management measures and working with Ontario Parks to install and maintain barriers, as well as erecting signage to increase compliance. So the recreational use compliance measures are also very important to reduce and mitigate impacts on species at risk habitat. The Conservation Reserve is also focused on protecting and enhancing species at risk and their habitat by undertaking surveys specifically for tree and shrub species at risk, and with a particular focus on American chestnut and eastern flowering dogwood. The Conservation Reserve have also located and will be controlling for various woody and herbaceous plants throughout the reserve. So just on this final slide here, I'd just like to bring your attention to the tree. The photo here was taken in the mid to late 19th century. Those are American chestnuts. I think the estimate was they were about 3 meters in diameter. We're lucky if we get an American chestnut that's 30 centimeters in diameter. So this is what our chestnuts used to look like before blight and poor harvesting practice took hold. So it would be fantastic to see trees like this again in our forests. So I would just like to thank all of the Forest and Tree Swamp partners. Every single person I work with is a pleasure to work with. I hope I can speak for everyone involved that we feel we're doing some very important conservation work in the LPWF Priority Place. It's fantastic to see positive on-the-ground action to make our forest and tree swamps healthier, not only for biodiversity, but for us bipedal primates as well. None of this work would be possible without the funding support from Environment and Climate Change Canada. I'd be happy to take any questions. I just wanted to mention that if you have a specific question for an organization, that you take down the contact information here and email them directly. But I'm happy to answer any general Forest and Tree Swamp Working Group questions, or especially Birds and Birds Canada, the work I do here. Thank you.
Thank you so much, Ian. I'd love to see the collaboration in all the groups here. And I'd love to see the action steps that you guys are doing. Very, very interesting. Let's open the questions. Anybody in the chat or wants to raise their hands and speak up? Michael had a comment. I think I will read this. It's pretty interesting too. It's a slightly earlier stage of development than the FRAG program, but we're also developing biocontrol of garlic mustard using a root crown mining, weevil, hoping to be able to scale that up and help with the garlic mustard problem in the years to come. So other groups working on this. My dog is talking. All right.
Your dogs have a question. You can let your dog know that the passive recreational use is included. He's included with that, or she's included with that.
I had a question, if that's okay.
Yeah, Jonas, go ahead.
What would you say is, looking at, since you've looked at all these different organizations, what do you think is missing or isn't done enough of so far? Or what could be, what do you think is a priority going forward?
I will speak on, what I think might be the priority, not necessarily what the group may think of the priority.
Of course.
So I won't speak to
I'm asking you.
for others. Yeah. So for me, it would be landowner engagement. I think that would be the biggest priority, especially being a forest bird biologist, engaging our forestry industry leaders, especially in Southwestern Ontario, and encouraging some either, if not best management practice of implementation, at least some level of habitat protection and reduction of unsustainable harvesting practices that still take place down here. So I think those are really things that I would like to see stressed moving forward. And I'm working towards that. Yeah.
We have another question I'm going to take over here because I think Emily is dealing with her dog.
No worries.
Brittany McLeod commented, "Thank you, Ian. Very well done. I loved how you summarized everyone's contributions and work."
Thanks, Brittany.
Yeah. Carrie has a question. Are the American chestnut trees in Bacchus Woods resistant to blight, or how have they managed to survive? I've been to many of the old growth forests around Southwestern Ontario, and I didn't see the American chestnuts anywhere else.
Yeah, and I don't know the biology too much about American chestnuts. I don't think they're resistant to blight. I think they have a protection buffer around them, being how big Bacchus Woods is. So I think that's kind of how they've managed to survive. But again, I'm not the foremost expert to talk about that tree disease in particular. So that would be my hypotheses if I had to answer that question.
Okay, thank you. I've got another statement here from Katrina. I really wish this amazing collaboration was celebrated in public. It would be great for my parents who watch the news rigorously to see this kind of information.
Yeah, absolutely. Yeah, absolutely. More than happy to put this information out to the public, especially as we get results coming through and see what all the groups have been doing in this priority place. It's absolutely fantastic.
Yeah, I think that's where we get our landowner integration happening there as well.
Sure. Yeah, absolutely.
One more question from Kathy. What tools were used to control garlic mustard? And can you
It froze on me. I see it here. What tools were used to control garlic mustard?
Yeah. Mm-hmm.
And can you see the extension of methods used to other sites?
Mm-hmm.
I'm not sure. Maybe someone can comment. Perhaps there's someone in the working group that that knows. It might be glyphosate, but I can't say 100% for sure. I think that's probably right now. I will say this very loosely, but probably the safest control measure at the moment. So I assume that would be used across other woody and maybe not woody, sorry, herbaceous plants as well.
Mm-hmm. Catherine says, "Hi, Ian. Thanks for this. As a private landowner, would love your insights on how to improve our wooded areas for SARS."
Yeah, absolutely. Feel free to email me. I'm working on best management practices for 2 of our species, the red-headed woodpecker and the cerulean warbler. Depending, of course, on your wooded area and what kind of habitat is already existing there. There might be some ways to improve and possibly encourage some species at risk. So yeah, please feel free to take my email address down and give me and send me an email.
Wonderful. A lot of good questions, and it seems that many people have an interest with regards to their own property. I've got one last thing that's popped up here from Michael McTavish, or it may have been physical removal. Yeah, we've done a lot of hand pulling along with the Norfolk cleanup crew. Yeah.
Yes, yeah, definitely.
Yeah. Thanks so much, everyone.
Thank you.
We will try and stay on time here. I've got one last research presenter, Jonas Hamberg, who we've heard quite a few very important questions that have come from him. He is a postdoctoral researcher at University of Toronto and with Toronto and Region Conservation Authority, with a master's in science in environmental science from SUNY College of Environmental Science and Forestry in upstate New York, and a PhD from University of Waterloo. During the PhD, Jonas collaborated with NCC at forest restoration sites north of Longpoint. Jonas' research is focused on restoration ecology, remote sensing, and thermal imaging. Jonas uses thermal imagery data from satellites, the International Space Station, and drone-mounted cameras to study how temperature changes as plant diversity and biomass changes over time in forests undergoing restorations or when affected by pests or disease. Welcome, Jonas.
Thank you. Let me see if I can get this working. Do you guys see the main
Yes.
part now?
We
Okay.
see your slides.
Do you still just see my slide?
There we go. You're set up.
Yep. Okay. Excellent. All right. As said, I'm Jonas. I work both with University of Toronto and TRCA. We're talking about thermal imaging for ecosystem conservation and restoration. This image right here is of me and others planting acorns in Thames Talbot restoration site about 5 years back under the auspices of Peter and Mary, who I think they were at least here in the morning. Maybe they're still here. So first of all, thermal cameras or imagers or bolometers or thermographers use specific materials sensitive to thermal radiation. The change in these materials is translated to temperature change. These cameras can be handheld or attached to drones, airplanes, satellites, or a space station. With decreasing prices and increasing accuracy, thermal cameras are being used by military, search and rescue, industrial applications, agriculture, and now ecology and conservation management. For ecology and conservation, it's mainly been used to track larger warm-blooded animals, as well as for measuring urban heat island effects. However, more cutting edge, or at least I think so, is to use thermal imaging of terrestrial vegetated ecosystem stress, for example, from disease, as well as to measure diversity and energy utilization. This is in part similar to agriculture, where thermal cameras are used to find stressed crops that need irrigation or fertilizer. But natural ecosystems, of course, are both more complex and more interesting, with different species, structures, and trophic interactions. So when we talk about climate change and the Earth getting warmer and the 2-degree goal, we generally think about air temperature as what we perceive as we walk around. But the air is in large part heated from the surface. Sun hits asphalt, roofs, fields, and forests. These surfaces warm up from the energy and then reflect or re-emit the heat outwards. When sunlight hits plants, they convert some of that energy to drive photosynthesis, transpirate water, and build more biomass. Some of that energy is also just held physically by the plant structure. This process cools the environment. The more biomass in the ecosystem, the more its surface is cooled. So when we restore successfully, we decrease daytime surface temperature. And when ecosystems degrade due to lack of conservation, they heat up. The more species of plants, the more it can potentially cool as well, as different species are better at photosynthesizing under different conditions in different places and in different seasons. Comparing a tree plantation to a more diverse forest, the hypothesis is that the more diverse forest will be colder during the day. It's more layers of vegetation, more leaf overlap, differences of root depth, and again, differences in daily and seasonal timing of activity. With thermal imaging, it may also be possible to spot stressed hotspots where vegetation is doing worse due to disease, drought, mismatch between species and the site, or any other issue, as these would come across as hotter. These hypotheses and ideas have so far gone mostly untested, especially in the context of restoration and when using thermal imaging and combining it with on-the-ground sampling of vegetation. So I use 2 different types of instruments, and each of them have pros and cons. So space-based instruments on satellites and the space station. The imagery is free and publicly available generally. You cover much larger extents, sometimes global. You get historic data going back as far as to 1984. The bad part is you have low resolution. Each pixel is somewhere between 60 and 120 meters for the best possible instruments. And clouds are a real issue. If it's cloudy, you're not getting a reading at that time. On the other hand, drones, or RPAS, as they are now referred to in Canada legally, remotely piloted aircraft systems, have high resolution down to centimeters. You decide when to fly. There's less error because there's less air or atmosphere in between the camera and the ground. However, they are expensive, although becoming cheaper, and it requires training, certification, approval, and time. So I've shown this actually at conference a few years back, but I will use it just as a background to talk about earlier work with NCC in Norfolk County. In very short, I use satellite thermal data to look at temperature change over time in NCC restoration sites. So this is Lake Erie Farms. Restoration was done in the bright green areas in 2006 by seeding around 100 different mid and late succession native tallgrass and oak woodland species, with emphasis in red and black oak. And I found that the summer daytime surface temperature decreased over 12 years from the beginning of restoration. I also found, when using vegetation data sampled on the ground by NCC staff, that this decrease was stronger in areas and plots with higher native biodiversity, even when controlling for total biomass and when controlling for non-native diversity. So this is temperature versus species diversity. So I'm going to move on to a newer study that is it's submitted but still in review. Here I focused on using one space-based thermal imager called EcoStress, and see if I can get this laser pointer thing. Spotlight? No. Anyway, it's the box in the complete middle of this photo that has sort of the arm attached to it. That is EcoStress. It's about 1 by 1 by 2 meters with a very advanced thermal camera inside it. It was launched and attached to the International Space Station in 2018. And the ISS and thereby EcoStress orbits Earth every 90 minutes on what's called a precessing orbit, which means that it shifts at which time it comes by each spot on Earth, meaning that we get images at different times of the day and night, which is different compared to most other satellite thermal cameras up there. So in this study, I had 2 questions. First of all, what is the relative difference in diurnal temperature, so day and night, over day and night temperature, and evapotranspiration, referred to often as ET, dynamics between sites in a pre-restoration state of agriculture, ongoing restoration, and forest sites similar to the goal state of restoration. Secondly, how does relative temperature and evapotranspiration change with the age of restoration sites? Can this change be modeled? So for this research, it took place in Southern Ontario, Canada, with the TRCA and in collaboration with NCC as well. And for this project, we looked at 2 groups of restoration sites, and I'll refer to those as the NCC and the TRCA group. There were a total of 43 restoration sites included. The majority of those were in our area here in Norfolk. Restoration was initiated at different years between 2007 and 2019. And the difference in initial year is what we used as a space for time substitution to look at ET and temperature differences. And to compare temperature and ET data taken at different times, what I did was to take the measurement for each site and then just subtract it by the measurement of a mature protected forest area in the same image. In this case, the mature in the NCC Norfolk area, I used a central chunk of Bacchus Woods for this. This is the most mature, most stable area, forest area. We use agriculture sites as the before restoration state, as most restoration was undertaken on former agricultural fields, and smaller forest sites as a post-restoration site. For the TRCA group of sites, we also included suburban residential areas. It's another common land cover type. So we found 19 usable ecostress LST images for the NCC area and 15 usable images for the TRCA area, all from 2020. And 9 usable evapotranspiration images only for the NCC sites. There just wasn't enough usable ones for the TRCA sites due to clouds, other issues with the equipment. And you can see here, this graph shows when each picture was taken. So it's over time of day and over date between June and September 2020. So it's a mix. So moving to the result. These are the results we found in terms of surface temperature relative to a large forest area with time of day grouped by site type. So think of the x-axis here, the black line, as the temperature for the NCC in Bacchus Woods, and then everything else is what is the temperature more or less than Bacchus Woods over the time of day. So if we look at the TRCA side of the graph on the right, we find that ex-urban residential areas were the warmest, the purple, about 12 degrees hotter at its peak than the large forest area. And it peaked around 2 p.m. Agricultural sites were the next warmest at 7 degrees more, and then restoration sites about 3, and then smaller forests about 2 degrees more. The NCC sites repeated the pattern, but were generally relatively hotter. I believe this has more to do with that Bacchus Woods by itself is very cool because it's such a large, intact, and sort of solid area. So it's not that NCC, the Norfolk sites are worse, it's that Bacchus Woods is just better. So to sort of think about this, if you're looking at Bacchus Woods on a, say, 20-degree day at 2 p.m., if you then moved yourself quickly into an agricultural field, it would be 30 degrees. On the restoration side, about 25 degrees, and the forest about 22.5 degrees. Same thing during the night, say it's a 10-degree night. It's going to be about the same in the other areas. It might even be a little bit colder, especially in the agriculture area. I also looked at the diurnal temperature buffering. So how big is the change over night and day? So I did that by measuring it as the standard deviation of relative surface temperature. And these were significantly different between all site types. For the NCC group of sites on the left, the mean standard deviation for agricultural sites was 5.2 degrees Celsius, 2.9 for restoration sites, and 1.7 for the smaller forest sites. And the pattern was the same for TRCA, but again, the residential sites, it's basically just how much do they vary over night and day. So you're seeing the forest sites just buffer temperatures much better. Here we looked at the relative land surface temperature of only restoration sites and splitting these into 5-year groups of when restoration was initiated. So red being the newest sites that had 1 to 5 years of restoration. Teal is 6 to 9, and then purple is 10 to 14 years old. And for the NCC site, there was a significant decrease in the temperature over day, and then especially significant in the middle of the day and afternoon, the older they got. So putting that sort of same data into what's called a mixed effect linear regression model, we found that per year, the temperatures decreased by about 0.1 degrees. This might not sound like a lot, but restoration takes time. And after about 32 years, the restoration areas would reach the same temperatures as the forest areas. So that makes some amount of sense in how long it takes to sort of restore a forest. Moving to instantaneous evapotranspiration. So how much water the trees let go of. Absolute ET decreased from midday to evening. Relative ET was marginally significantly higher for agricultural sites compared to forests. One surprising finding was that relative evapotranspiration actually decreased significantly by years since restoration in the NCC sites. Overall, the newest restoration sites had an ET that was around 12 watts per square meter, higher than forests. So here, it doesn't make a direct sense, but here evapotranspiration is measured as a unit of energy, how much energy it takes to transpire that water. So seems strange, but this could be due to more exposed soil and thereby higher soil evaporation in the newer sites. And over time, these sites become more like the forest in terms of evapotranspiration. Excuse me. So we find that EcoStress provides temporally denser data than Landsat, higher resolution than Landsat is not a satellite, higher resolution than MODIS satellites, which is another type of satellite. But there are still limitations with sites that are smaller than 5 hectares. Summer diurnal temperature variation decreased, i.e. thermal buffering increased substantially by restoration. And we believe that thermal buffering can be quantified as an ecosystem service by EcoStress and similar instruments. Monitoring restoration using EcoStress and similar instruments is possible, but requires an easy-to-use service where managers can check the temperature and ET trajectory of each site once per season. I'm kind of dying a little bit over here of my coughing, so I'm gonna want to show you this video if I can, but I'm not going to provide too much detail. But just to show you, this is one of my research sites. This is what it looks like when you're using a drone. So I'm also using drone for my research. Here we're looking at what's the temperature impact of topsoil transfer. And while I use straight down still images, even in this video, the forest is a cooler yellow, while the agriculture or former agriculture area is sort of an orange that is hotter. And then you see the road that's sort of pink, red. We'll come back again. Yeah, so I am not going to cover this more than that. When it was a topsoil transfer experiment for restoration, we found that if we transfer topsoil from a forest to a field, we increase native species diversity, and we also decrease temperatures significantly over time. And that the temperature correlates most strongly with native forest species richness, more so than tree height, tree diameter, or all species richness. I'm right now working with RCA, and some of the things I'm looking at is to quantify and map thermal impact of leaf defoliators and other tree pests. And I'm looking at how biodiversity, complexity, and fragmentation impacts forest LST, land surface temperature, using both drones and satellites. So here's just an example of what I'm doing right now. It is the effect of LDD moth or Limantria dispar, which was usually before known as gypsy moth. This year, as I'm sure many of you know, the outbreak was very severe, so I wanted to see how it affected temperatures. So I flew over a number of sites in the GTA area with thermal cameras, and here you can see on the left some poplars heavily defoliated, as well as sort of more unaffected trees. And you see a mean difference of about 3 degrees in temperature. So I wanted to see if, can we map it? Can we find it? What's the impact? I also flew over sites that have been treated with BTK spray, and we're going to see if that did anything. So that's aside. Also looking at the Meadowway project, which is a meadow restoration under power lines in Toronto and Scarborough. Again, looking at the sort of cooling effect, seeing that meadows and shrubs cool much more than lawn, unsurprisingly. And then I'm looking at if we can use other instruments as well, including this GIF is of me using a LiDAR 3 days ago. LiDAR is a technology that shoots hundreds of thousands of light beams per second at the ground and measures how long it takes to reflect in order to create 3D images of the terrain. And together, LiDAR, thermal imagery, and multispectral cameras, which I also have, could help us map and hopefully pinpoint colonies of invasive species, including those of dog strangling vine, which is a big problem for TRCA. Finish off, I want to set this work and its importance and potential applications in a wider context. We have hot days ahead. It's going to get hotter. We're going to get probably an extra week of 30+ degree days by 2030. This kills people, this kills, stresses, and displaces animals and pets. It also helps spread diseases and invasive species. However, I think that we have an opportunity in thermal imaging to show off natural cooling centers, such as parks, ravines, forests, even street trees, and meadows and wetlands. I think I haven't gotten to wetlands. And then just as a sort of reminder with COVID, cooling centers, especially in cities, cooling centers such as libraries were not always accessible, and they're not always accessible even generally if you're not close to them. So nature is more important than ever to people in order to cool off. I believe this is important for planners, that they can locate heat deserts and hot watersheds for restoration planning. Managers can monitor restoration success and biodiversity based on temperature. Grad creators can show funders how your restoration decreases temperatures. The public can think about, are you in a heat desert? Are your neighbors or someone you care about? How far does someone have to walk to get to a cool green space on a 30-degree day? How will new development coming up affect that? Will it make it worse? And in that, advocate for plant more native and diverse greenery, and protect parks, street trees, all natural areas as cool islands. With that, I would like to thank co-authors, my many research assistants, super helpful collaborators, including many who are here and listening, as well as all of you who attended. Thank you. And if you're interested in more, you can email me or check out my current paper in ecological indicators and hopefully upcoming paper in remote sensing of environment. Thank you.
Thank you very much, Jonah. You survived this. Thank you for pushing through and sharing all this amazing work that you're doing. We are pushing our time a little bit, but let's see if we can get a few questions from the group. Again, feel free to chat it up or speak up.
Jonah?
Yeah.
Thinking that locally we have different types of forest. Your
Mhm.
comparisons seem to be between forest and other vegetation zones. Have you done studies between different types of forest? I'm thinking that locally we have still some leftover red pine plantation. We have mixed deciduous pine, we have dense deciduous, and we have some red oak savanna. Have you ever done that kind of hot day drone study to compare the heat between
No,
the types of forest?
yeah, it's a great question, and it's basically what I'm starting to do probably in the next month or two. Unfortunately, not in the Norfolk area because TRCA is paying me, but basically I have their ELC data, their ecological land classification, and that's exactly what we want to do. We want to look at, okay, what's the difference between plantations and more natural forests? What's the effect of sort of the complexity, the age of the forest, the species composition? That's what I'm going to be doing for the next half year. I didn't really get into that because I haven't started it, but yeah, I'll be using satellites for that because the drone just doesn't cover enough ground. But yeah.
As an aside, a lot of studies have been done on that specific issue in Northwestern Europe and in Scotland. And they might be interesting studies. They found in Scotland where they did pine plantations, the pine plantations raised the temperatures even more than non-forest areas.
Interesting.
I thought it'd be interesting to see how that works.
Yeah. Is this Smigaj? Because she did some studies. I don't know if that was her or if that's someone else.
Sounds familiar.
That's the only one in Scotland I know, but if you have that, please send it to me if you can find it.
Okay.
Yeah.
Thank you, Mr. Gibbons. Great questions there. We have one more in the chat from Elizabeth. How did you deal with the spatial scale? Did you average pixels to get a value per site? I figured that the spatial resolution of eco-stress may be not good for working in small patches of forest. What do you think?
For sure. I had to, so the smallest sites I worked with were about 2 hectares. So that was, so the pixels from eco-stress is 70 by 70 meters. So it's like, 4 or 5 pixels per site. That's really too little. And that's kind of what we found. Like if you're going on a site that's smaller than 5 hectares, that's after I actually buffer each area. I don't take the edge. I take 50 meters off the edge because the size of the pixels, and then I take the average for the site. But that's basically that's the drawback of the space-based instrument compared to the drone, is just the size of the site. They have to be a certain size.
Great. Thanks, Jonah. Great question, Elizabeth. Any other questions on this last presentation? All right, back over to you, Cynthia. Thank you, Jonah.
Merci beaucoup. I'd like to introduce to you Brittany McLeod, a habitat ecologist with CWS from the Ministry of Environment and Climate Change, to do a wrap up before we thank all of you for being present at this conference. Brittany, welcome.
Hi, thanks, Cynthia. Let me just share my screen here. Don't have a lot of slides, but just some visuals. Oh, geez, my terrible, all the files. Okay, that's not what I wanted to show you. All right. Okay, you can hear me, you can see me, everything's good. All right. Hi, everyone. My name is Brittany McLeod. As Cynthia said, I'm an ecologist with the Canadian Wildlife Service. We're a branch of Environment and Climate Change Canada, and I coordinate the Priority Place Initiative in Ontario, which I know you've heard bits and pieces about during this conference and in past conferences. This has been awesome. I've enjoyed listening to all of the great talks about your conservation research and work happening in the Longpoint Biosphere region. It's truly inspirational. As you all know, it's a very special place, and the collective impact of our work, whether you're a researcher, land manager, wildlife technician, biologist, policy analyst, local activist, or community advocate, is making an important difference on the landscape for wildlife and ecosystems found here. So Cynthia and Ian both gave a bit of an introduction to the fact that a portion of the Longpoint Biosphere region in Norfolk County is a federal and provincial priority place in Canada. But what does that mean, and how does it relate to what was discussed today? And what have we been doing? So priority places are part of the Pan-Canadian Agreement on Species at Risk Conservation. So this is a federal and provincial and territorial agreement for species at risk. And so it's one of the pillars of how the governments have said that they will work together to further conservation for species at risk. And so priority places seek to conserve Canada's biodiversity by advancing action on the ground in a collaborative way, focusing on multi-species and ecosystem-based approaches. And so since 2018, we've been working to put this government agreement into action with tangible results on the ground. And so we've been working with the local conservation community in Longpoint Walsingham Forest to develop and implement this integrated conservation action plan that Ian talked about today to guide our efforts. And so we really utilize the local research and knowledge to develop what the priority strategies in those plans are that address key threats affecting ecosystems and species we're trying to conserve. And many of those threats were touched on today, including invasive species, agricultural runoff, roads, fire suppression, in tall grass communities, and logging and wood harvesting. And so we've come together in a collaborative way to join forces to become more coordinated and effective in advancing positive change for conservation in the region. This has included taking an adaptive management approach and involves looking at the entire situation as a system and as a whole. I realized I did not show any of my slides here, but I just was reading my notes. Anyways, so I wanted to put up this slide that has a whole bunch of results displayed on there, some outputs from the initiative so far. We're working right now to develop a more fulsome progress report on the implementation of the ICAP, but this is kind of like a nice visual of really high-level summary results of work from the different working groups that have happened since 2018. And it's the result of a significant effort and hard work from the local conservation community. So many of the organizations I'd like to acknowledge here on this slide. It's been truly amazing to see what's been achieved when we come together as a community to affect change on the landscape. We may come from different perspectives or organizations with differing mandates and agendas, but in the end, we have a common goal that we're trying to achieve, and we're so much more effective together. So I know it's approaching the end of the day, and I don't want to take too much of your time. I'd encourage you to find more information on this initiative and the work being done in the priority place on the website listed here. It's also linked through the Biosphere Region's website, lots of information there. And you can email me directly to discuss any initiatives or actions further. So in closing, I'd like to thank the conference organizers for bringing us together today and all of the speakers for their interesting and inspirational talks, as well as everyone in attendance for your support of the Longpoint Biosphere Region Annual Research and Conservation Conference. We hope to see you all again next year, and thank you and take care.
Thank you so much, Brittany, for that wrap up. That was a good way of pulling all of this information together to make it fairly clear on the fact that collaborations are so important right now. I want to thank Amelie for being there and doing all the tech work and housekeeping that I can't do when I'm doing other things, and all of the people that joined us to listen into these presentations that were very, very interesting. There's room for questions. I see a couple red highlights there. Oh, it's all people saying thank you very much for a wonderful conference, basically. And I look forward to hosting this again next year and bringing even more updates to you from our working groups here in the Walsingham Forest Priority Places and on behalf of the Longpoint Biosphere Region. Thank you so much.
Just going to put one little note before I let you go. It's been a pleasure to have you all today. And for the Longpoint Biosphere, if you feel like you want to get involved, and there's different ways, and I hope that we have the opportunity to collaborate, partner together moving forward. So it's truly an inspiring day. Thank you, Cynthia, for leading the way, and thank you to all our amazing guests. I will be posting the replay of this conference. I will send you an email if you missed any part of it, and we look forward to seeing you next year. Thank you so much.